CRISPR is something that many of you may have heard of, but might not know too much about. It is a genome editing tool that is part of a bacterial immune system created to defend against viruses. So here's a bacterium with its flagellum and circular DNA. Like many things in biology, CRISPR was named before anyone really knew what it did, so the name might not seem to fit the function here, but it stands for Clustered Regularly Interspersed Short Palindromic Repeats. Right, CRISPR is much easier to remember. But basically, CRISPR is a repeated sequence of DNA, which I'm showing with these red dots along the chromosome. Alright, so how does it work? The CRISPR system has two parts. One is an endonuclease protein called CAS9, where CAS just stands for CRISPR-associated system, and the other is a guide RNA that is complementary to a specific DNA sequence. Both of these are encoded within that repeat sequence on the bacterial DNA. So the gRNA is loaded onto the Cas9 protein and creates this CRISPR-Cas9 complex that will encounter a viral DNA, which it could then cut. Now this is not just any bacterium, but it's one that has evolved to be resistant to this particular virus, and the mechanism of this resistance is a gRNA being complementary to part of the viral DNA, which gives the system specificity. Here's an image straight from your first aid book. So remember, we have the guide RNA here, and the Cas9 endonuclease, which is this entire blue structure that you see in this image, and together they form the CRISPR-Cas9 complex. Now this complex is able to locate and bind to a specific sequence of genomic DNA, and this sequence is called PAM. Once bound, the guide RNA can unwind a specific area of DNA, and again, this is because the guide RNA is complementary to this specific area. Cas9 can now cut the DNA and create either a single or double-stranded break. Now our cells naturally try to fix this break by non-homologous end joining, but is not successful in doing so. This results in accidental frameshift mutations, which can essentially knock out and disable gene function. Additionally, donor DNA sequence can be added to fill in this gap using homology-directed repair. Now, what can we do with CRISPR-Cas9? It's easy to imagine how we could modify this system by changing the sequence of the guide RNA to whatever we want, which would allow us to target any DNA sequence we want. For example, we could create a guide RNA sequence that's complementary to the area around a specific area of the CFTR gene. Do you remember what the most common mutation is that causes cystic fibrosis? Right, it's a deletion of the 508 codon of the CFTR, which codes for phenylalanine. Now, if we want to edit the sequence, we need to introduce one more element to our system here. We can add another sequence of DNA which is complementary to the gene of interest, but with the correction we want to make. So in this case of a missing codon in the CFTR gene, we can introduce a new sequence which isn't missing anything. When the cell's native DNA system arrives to fix the break made by Cas9, it can use this new sequence as a template. And now we fixed an unwanted mutation. Pretty cool, right? Now CRISPR-Cas9 technology is very theoretical right now. It has been proposed as a treatment for multiple human diseases, especially those with a genetic cause. However, studies and applications are still very new, meaning human research and data is still up and coming. However, research in animal models have been done and suggest CRISPR technology will have the potential to treat an array of diseases, including different cancers, blood disorders like beta thalassemia and sickle cell disease, cystic fibrosis like in our example, Duchenne's muscular dystrophy, and even heart disease. The future seems pretty bright for CRISPR. And if you take any time away from your studies to listen to NPR or to watch the news, you may have heard CRISPR-Cas9 recently. A scientist in China used CRISPR technology to modify one-day-old embryos' CCR5 gene, the gene that enables HIV, to enter and infect our immune system cells. He then implanted these embryos into a woman's womb, and this resulted in a viable pregnancy and the birth of two twin girls, both free of HIV. The father in this situation and donor of the sperm was HIV positive. Now this research and experiment has been the face of controversy since news broke out, and the scientist is currently undergoing review by an ethics committee. But whether you agree or disagree that this is unethical and unnecessary, you have to admit it's pretty interesting. Thanks for watching, and if you liked the video, give it a thumbs up.